Okay, I want you to picture something. It's the 1960s. Over here, you've got biology, labs, microscopes, petri dishes, and way over there, you have high-speed computing, these massive rooms filled with gigantic tape-driven machines. These two worlds, they might as well have been on different planets. They barely even spoke the same language. But one scientist, well, she saw a future that almost nobody else could even begin to imagine. And that brings us to the central question, the big challenge. How in the world do you take the complex, messy, beautiful stuff of life and turn it into something a computer can actually read, index, and search? The answer, it turns out, came from Margaret Dayhoff, a true pioneer who had this vision of a world where the secrets of life could be unlocked not just with a microscope, but with a computer. You have to understand just how radical Dayhoff's vision was for her time. She was proposing that we apply the cold, hard logic of computing to biological sequences. The very building blocks of proteins. The idea was to create a library, a database of life itself that scientists could systematically analyze. This wasn't just some new idea, it was literally the start of a whole new science. So how did she pull it off? Well, it all started back in 1965 with the publication of the very first Atlas of Protein Sequence and Structure. Through the 70s, that atlas grew and evolved into what became the Protein Information Resource, or PIR. And by the 1980s, the principles she laid down became the absolute bedrock for the massive global databases we all use today, like GenBank. Okay, let's zoom in on that first incredible achievement. When you hear the word atlas, you probably picture a book of maps, right? And honestly, you're not wrong. In a way, that's exactly what it was. A map of the entire known protein world. But it was so much more than just a book you could flip through. The design, that was the secret sauce. The atlas was the very first comprehensive computer-based election of its kind, and it was built with a revolutionary purpose, to be a structured, searchable archive. That's what changed everything. It transformed a static collection of information into this dynamic tool for discovery, and it laid the direct groundwork for the PIR. But here's the thing. To build this archive, Dayhoff quickly realized she was going to need new tools. The language biologists were using just wasn't going to work for computers. So, in what has to be one of the most brilliant and practical moves in the history of science, she just invented a new one. She created the one-letter amino acid code. It was a simple, elegant, and totally standardized symbol for each of the 20 common amino acids that make up proteins. This system was so incredibly effective, so intuitive, that it's still the universal standard in biology and genetics today. Think about that. And this shows you just what a huge leap forward it was. Before Dayhoff, scientists used these three-letter codes, like ALA for alanine. It was so cumbersome and clunky for computers to handle. Dayhoff's innovation turned it into a simple A. This new language was just elegant, simple, and, most importantly, perfectly machine-readable, a total game-changer. But she didn't stop there. Oh no, she also developed what are called point-accepted mutation, or PAM matrices. This incredible tool tackles a really fundamental question. How related are two different proteins, really? PAM matrices calculate the probability of one amino acid changing into another over evolutionary time, which lets scientists compare sequences in a truly meaningful way. So let's connect all this to right now, today. We've been talking about her early work, but the true measure of her genius is how that work, from way back in the 60s and 70s, is directly powering the science of the 21st century. This quote just nails it, doesn't it? Of course, by modern standards, the computers she was working with were ancient, her systems were absolutely primitive, but her vision, that core idea that biology could be indexed and compared and understood through computation, that was utterly radical. She was decades ahead of her time. Seriously, her foundational work powers pretty much every corner of modern biology. You know that sequence alignment tool, BLAST, that millions of scientists use to compare genetic code? It relies on her principles. Those giant global protein databases like UniProt, they're direct descendants of her atlas. Any research into evolution, genetic diseases, molecular modeling, all of it stands on the shoulders of her work. When you really step back and look at the big picture, her contribution was so much bigger than any single tool or database. What Margaret Dayhoff really did was create an entire scientific discipline from scratch. And even though she passed away back in 1983, her influence has only gotten stronger. This isn't some historical footnote. It is a living, breathing legacy. Her work is right there in every modern biology lab, it's embedded in every genome sequencing project, and it's the engine running behind every single protein search a researcher performs. And that, right there, is the perfect way to sum up her life's work. She didn't just build a database, she built a field. By creating that first database, she gave scientists the tools they needed to invent an entirely new way of doing science: bioinformatics. She fundamentally changed how we study life itself. Her story leaves us with a really powerful question for today. Dayhoff saw this incredible untapped potential in combining biology with data science. And it really makes you wonder, what are the next great frontiers? What future discoveries will be unlocked when we apply that same radical vision to combine data with chemistry or neuroscience or even ecology? The next revolution is out there, just waiting for its own pioneer.